Okay, so today we're going to talk about and discuss about this PyMol. Okay, so let's start the class. Okay, so the last class we saw actually how we can model the particular protein, but obviously after model, the thing is that we have to visualize the protein in a great detail, how the entire protein is behaving, how the amino acids are pointing, how they are placed around the core and all. Okay, so the basic thing about PyMol is first we have to understand how can we use it for movement. Now, always advise that for using PyMol, always use a mouse. Okay, mouse gives a complete freedom and it gives easy access to use this PyMol. So first the left click. First, if you need to zoom in and zoom out, all you have to do is click, hold the right click button, and then you can move the mouse up and down to zoom in and zoom out. Just hold the right button, and then simply zoom in and move the mouse up and down, and it's going to zoom in and zoom out. Okay. Then to rotate the entire molecule like that around the central axis, all you have to do is hold the left mouse button. All you have to do is hold the left mouse button, and you can move the mouse to rotate the entire protein in a central axis. Okay, and the middle mouse button or the mouse wheel allows you to translate the molecule. If you need to translate from this side to that side, then that you can do using the middle mouse button or the mouse wheel. Okay, so that's the basic essence of how to use the mouse for the movement. Okay, so that is why it's good to have a mouse, because only using the laptop keyboard doesn't give you full freedom of movement of this entire thing. Okay, Acha. So now the first thing that is that, so this is the protein, but as you can see, this is only the backbone of the protein. We can't visualize the entire amino acids. The amino acids are not visible, the side chains are hidden, only the backbone is present. So to see the backbone and all, or to see the there are various options to visualize a protein. The first option is actually, so for that we have to use these following keys, the ASHLC that are present in this right hand corner. Okay. So the first thing that it does is, first let me hide everything. Okay. So S is for show, H is for hide. S and H are antagonistic one another. Okay, whatever the S shows you, the H can hide that particular thing. Okay, so first the option is lines. As I click on S, you see the entire parody opens up, and then the panel opens up, and then the first option there is lines. So if I click on lines, then entire amino acids, everything will be visible in the mode of lines. But you see here, you can understand the structure of the amino acids. The structure of the amino acids are visible. But one thing is lacking is that you can't understand the structure. The secondary structure you can't understand. Correct. The secondary structure is not visible to you also. Okay. So that is one drawback. But obviously you can visualize the entire molecule, the everything, the amino acids and all. You see, this is the amino acid and all. Okay. So, but these are lines. So what the other options? So to hide lines, what you can do, you can again click on H and click on lines. So it's going to hide everything. After hiding the lines, there is another option, that is sticks. The sticks are nothing but thick lines. Okay, the thick lines. So you see, this is the stick model, where the lines are very thin, sticks are really fat and all. Okay, so it gives you a better adjustment to understand that, show the structure.
Sir,
Yes.
there is someone at the door. Can you excuse me for 5 minutes?
Yeah, sure. Go.
Thank you, sir.
Sure.
Yes, I am back.
Okay. Okay. So this is now obviously to hide that particular lines. You can do is click on H and click on sticks. It's going to get hidden. So after lines, next we have is the ribbon. So what is ribbon? Ribbon gives you a faint idea about just the backbone, which also gives you fairly good idea about the entire structure. Okay, as you can see, this is the alpha helix because the ribbons is actually packed in like that. It's alpha helix, and these are the other things. But it is really difficult to extract out more information from the ribbons. So generally, we don't use that. It's always better to use cartoon. Okay, cartoon is the method which gives you the best representation of your backbone, be it alpha helix or beta sheets or these loops. It gives you the best representation. Okay, and on top of that, what you can do, you can simply mix and match also. For example, let me hide the ribbons first. Suppose since the side chains are not visible, what I can do is I can simply go and click on lines. So if I click on lines, I have the background, I have the backbone, and also I have the amino acid side chain as well, and I can see in which direction the amino acid side chains are protruding. Okay, is something protruding inside or protruding outside? Those are the things. But one thing that you can see is you also have to learn about the sequence of the amino acid, right? Which sequence is that? For that, there are two options. One thing that you can do is obviously that you can simply right click on a particular option. If you want to understand what this amino acid is, right click on that option and you can see the amino acid number. That is, it is glutamine 2. It is glutamine 2. But again, what's the easier option? The easier option is, see, in the same panel, if you come down, then there's a playlist option, like you can play, and then there's an option called S. If you click on S, then you see the sequence panel opens up on top of your structure. This particular panel on the right hand side, on the lower right hand side, on the lower right hand corner, you can see there's a panel called S, and you can click on S, and then the entire panel opens up, the sequence panel opens up, and then you can see the sequence. And what best is that whatever sequence or whatever amino acid you select over here, those amino acid get selected in the main screen also. As you can see, suppose I am selecting only this 4, so see, this 4 amino acid got selected. Again, if I am clicking something over here, suppose I am clicking over here, you see, I select the tyrosine and see the tyrosine got highlighted. The 8th tyrosine got highlighted, and you also can see the number of the amino acid. The upper, this number represents the number of the amino acid. So suppose this 6 means this is the 6th amino acid. 11 means this is the 11th amino acid, like that. Okay. So suppose you want to select only the alpha helix. So like that, I can select the alpha helix. So this portion builds up the alpha helix, not the last one. So suppose this portion builds up the alpha helix. So I can select alpha helix like that. Okay. Or if you want to select or mutate or simply check any residue from there, you can also do that. Okay. Okay. Now, suppose now generally what happens is that in our body, the protein does not stay like this. Whenever we crystallize a protein or we visualize a protein, the protein does not is not like this. You can't see through the protein. Here, what I can see, I can simply see through the protein, right? It's like this protein is extremely transparent to me, but that doesn't happen. So, how does the protein behave in our body? The protein stays as a 3D object. For example, you take your bed. If it's a divan bed, then obviously you know that the inside is hollow. But can you see through it? You can't. We have to
No.
see the entire structure, the 3D structure. The same thing goes for the protein also. We can't see through the protein. Okay. So how to know the 3D topology? How would I know how the 3D topology is looking like? For that, again, we have to go to S. Now there are two options for that. First, we can click on mesh. See, if I click on mesh, then what happens? Then you get the idea about the 3D topology. This is how the protein actually looks like in the body. This is how the protein actually looks like. Okay, but still you can see through it, but generally it simply gives you the idea about the secondary structure. Also get to know the secondary structure as well as the 3D topology. So this is how the protein actually behaves. But to actually understand how the protein looks at the 3D surface, then we have to click on this surface option. The surface option generates the entire surface topology of the protein. See, this is actually how the protein looks like in our body. This is the entire thing. What is this color codings? You can see multiple colors, right? What are
Yes, sir.
these colors?
So maybe different amino acids.
Not
To be
amino
honest.
acids, but different elements. See,
Okay.
the red indicates the oxygen atoms. Okay, that means it is electronegative zone. The blue represents the nitrogen atom, that is electropositive elements. The yellow is actually the sulfur. Now, which amino acid contains sulfur?
Sir, I remember. Just a second. There is methionine.
Cysteine. Okay,
Cysteine.
cysteine and methionine. So,
Methionine.
yeah, so
Yeah.
this means either there is a cysteine or methionine. You have to check what is that. If I'm clicking over here, yes, there is cysteine. Okay, so yes, there is cysteine. So like that again, and this green indicates the carbon atom. So this is how actually the protein looks like. Now, what is the utility of the surface? The utility of the surface is that not only gives you the best view of the protein, how it looks like, but it also gives you the idea about the ligand binding site. You seeing these crevices? Can you understand these crevices? This
Yes,
hollow
sir.
zone,
Like
the
there is.
crevices. These
Yes.
crevices are actually the ligand binding zone. This crevice over here. These are actually the ligand binding zone, because this gives the ligand. If you have a ligand or if you have another protein that is binding to your protein interaction, protein ligand interaction, then these are there where the other protein or the other ligand docks to. These are the docking zones. Okay, these are the docking sites and docking zones. So this is actually the best. Let me hide the mesh. So this is it. But again, I'm telling you that one thing. But here you are only getting the surface. But we can also get to know the electronic ensemble of the protein, because there are certain zones. Though I have told you that this red zones indicates electronegativity or electropositivity, but you can see there are certain zones where the oxygen atoms are rich in number. For example, over here you can see the reds are lot in number. That means this particular domain is electronegative, highly electronegative. But is it only this domain that is electronegative? Is it only this domain that is electronegative? For
No, sir.
that, there are
Other
other
domains
zones.
are.
There are other zones also, right? Exactly. But there are also others. For example, this particular, it is also electropositive zone, the blue region that is indicating. But if you are having a lot of red or electronegativity over here, then the electropositivity kinds of gets lost. So to understand the entire electro shape of the protein, the entire electronic assembly, we can also use the vacuum electrostatics to generate the electrostatic of our protein, because protein generally uses electrostatics, van der Waals interaction to interact with other protein or other DNAs. Okay, if it's a transcription factor, then it uses the electrostatic to bind to a DNA. Okay, I'm showing you how. So first, let me hide the surface. Okay, so this is it. Let me hide the lines also. Now, one other thing is very important. That is how to color. Now, see, as I am indicating, let me first. Yeah, see here you can see these color codings. The red indicates oxygen atoms, the white indicates hydrogen, the blue indicates amino group. So that means over here, what is this? NH3 group, nitrogen and 3 hydrogen. That is NH3 group. This is oxygen and OH. That means it's a OH group. Like that you can understand. But you can also change this color. How to change the color? See, you can't change the color of oxygen or nitrogen. The color of oxygen, nitrogen, hydrogen is constant. All you can do is change the color of the backbone. How you can do this? See, there is a color option over here, C, right? C stands for color, and you can see this is actually color coded. So what you can do, you can color it by element. First is, as I open it up, there is colored by element. So if I click this, see, then all I am doing is changing the color of the backbone to magenta. All of the other things are maintaining their color. Okay. Similarly, you can also change the color to like this, whatever you feel like. Whatever you feel, you can change the color to like that. It depends upon your choice. Okay. Or you can also go by a coloring in secondary structure. What is that? See, in secondary structure, if I color it by secondary structure, then every secondary structure assumes a different color. For example, you see the helix are assuming a red color, the beta sheets are assuming a yellow color, and this is a slight helix, it's also assuming yellow color, and the loops are in green color.
Green colors.
So like that, you can also color coded by secondary structure. There are also multiple color coding available. Now, why I'm color coding it? Because sometimes the secondary structures give you a very good answers. Because the regions which is secondary structure, that particular part is highly stable. Highly stable. The amino acid present in the loops are not that stable compared to the amino acid present in the helix, because they are simply, since they are in a secondary structure, they are going to maintain their structure, and this maintaining of the structure is very constant. So even if you add some HCl and all, the loops are the first that is going to get troubled, and secondary structure regions are the later, the part which is going to get unstructured in the later part when you increase the concentration of HCl or you increase the temperature. Okay. Again, you can also color it by a single color. You have red and you have different shades of red. You have different color. You have sand, you have tint, whatever color you want, you can color it. But the thing is that if you color it by in a single color, then everything gets colored. See, everything gets colored. You are not understanding, or you can't color code it differently. Only if you color it by element, then only you can get to see the different colors of the elements. Okay. But if you simply move into, but these are actually required, because suppose you want to give a picture to your thesis, you want to upload a picture to your thesis and all, then of course you need good representations. Then this particular color coding allows you to give a good representation. Sometimes there are two molecules. Suppose you are seeing a docked structure. When I'm seeing a docked structure, then obviously coloring two different proteins into different manner ensures that you can understand which two proteins are that. Okay. So like that you can understand and see the structures like that. Okay. Now that the thing that I was talking about is that one more thing to discuss over here is that why it is important to see the amino acid, because you see over here, when I am, let me highlight just a minute. Just a minute. Let me highlight this entire stretch. Okay. Now after. Okay. Now you see over here. Let me color it differently. Okay. Now you see over here. In this particular beta sheet, there are few amino acids that is protruding outside, right? Like this amino acid is protruding outside. As you can see, this is the lysine. This is protruding outside. This particular molecule, that is the asparagine, is protruding outside. This molecule is protruding outside, that is valine. Whereas this particular part over here, the isoleucine, is protruding inside. Whereas this is also isoleucine. This is also protruding inside. Now, what is the difference? The major difference in the protruding inside and outside is that the molecules that are protruding inside, it is present inside the protein. So these amino acids are not there for protein interaction or ligand interactions. Only the amino acids that are protruding outside, they have the capacity to interact with a particular part. Because if you simply check, see, this is the part that I colored in blue. Okay, so see, let me color it in a different color so it becomes easier for you to understand. Suppose yellow. Now see, in this particular part you can see that this is actually the lysine, or the lysine that was protruding outside. So that means these are the residues that is going to interact with the ligand or a protein. Whereas the other residues, the residues like isoleucine and this, these are all inside. You can see these are inside actually. These are not protruding outside. These are actually inside the molecule. Okay. Since these are inside the molecule, since these are in, see, I have color coded them to here, and now see these are inside. So these are actually the other isoleucine. One isoleucine is still on the surface, but the other isoleucine, see, it is, you can't see that on the surface because that is not present on the surface. This is actually protruded inside. So that means this particular isoleucine is not required, and this is actually not required for any interaction. So if for a particular protein, if you find out that this particular isoleucine is mutated, then it is least likely to inhibit the functionality of the protein. Why? Because it is not present on the surface. And the surface amino acids are the major amino acid that participates or that dictates the function of a protein. Because that is the one which is interacting. For example, our hands and our legs are the major thing that is doing every day-to-day job. Yes, there are other portions like your heart, kidney, all are actually required for day-to-day activity. But can you hold anything with your liver? Nothing.
Sir.
The liver doesn't have that kind of function. So in case of the structure function relationship, the SAR, that is structure function relationship, structure activity relationship, in that portion, the isoleucine has no effect. So this is the utility of this structure identifying, that which amino acids are most important for this particular protein and which are not. That kind of analysis. Okay. Again, as I was talking about the electrostatics, that as you can see that yes, we understand the electropositive, electronegative element, but what about the electrostatics? For that, we have to go to the A option. In the A option, there is an option called generate. Okay. Under generate, there is an option called vacuum electrostatics. So if you simply click on, then under vacuum electrostatics, there is an option called protein contact potential. I'm again stating you, click on A. The A button stands for action. Okay. Under action, there is an option called generate. Under generate, there is an option called vacuum electrostatics. Under vacuum electrostatics, there is an option called protein contact potential. So if we click it, then it's going to show you the protein contact potential of your molecule. Now this is actually the entire thing. For example, as you can see, this blue indicates the electropositive zone. The greater the deep the blue, the more electropositive is it, and greater the red, more electronegative is it. So you see in this particular domain is highly electronegative. This entire domain is highly electronegative. So if you have any other protein which is electronegative, then that particular part is not going to interact with that. Okay. So here over here only some highly electropositive entity is going to interact with this particular domain over here. Okay. Whereas this part and these zones are electropositive zones. So this gives you an idea about the probable docking site of the protein. So if you have two proteins and you know which proteins look like, what is the activity of the protein, or if you have a protein transcription factor that can bind to the DNA, we know that the DNA is negatively charged. So of course the DNA, if this was a transcription factor, which is not, if this was a transcription factor, then it would not have bonded to the DNA using this particular part, because this is electronegative. Rather, it would have used this domain to bind to the DNA, as it is electropositive, or this domain. Though this is not a very entire, the domain is very short, but still. Okay. So this is how you also can understand which are the probable docking site of the protein, and if it's a transcription factor, which is the docking site of the DNA also. Clear? And the
Yes, sir.
best, most important part of that, you can also take pictures. For example, suppose I want to take a picture of this. Suppose I want to state, yes, this is the structure and this is the part. So simply arrange the molecule in whatever you way want, and then simply go to files, and then simply click on export image as PNG, save PNG image as, and then simply save the image, and simply give you a random name, and then this particular molecule gets saved in your computer. See, the molecule gets saved. Okay, the image gets saved. So this is the other way where you can save the image, because you can obviously use this up in your thesis on your whatever, visualize and all. So this is the other part. This is why you also require this particular thing. Even if you don't want to do anything, then also you actually require it to set the image. You actually require it to save the image so that you can represent it, show it. Okay, that kind of thing. Okay, so any question?
Sir, I
Yeah.
just have a question.
Yes.
Like yesterday, I was trying that Roberto model that you have shown and the website and
Yes.
the link that you have provided.
Yeah.
Sir, from where we need to take the protein structure? Like, because if it from NCBI, can you show me once?
Yes, surely. See,
Yes.
for protein structure, it's better not to. Yes, you can go to NCBI. All right. Just wait a minute. Simply open the NCBI and then click on this particular database to click on protein. Select the protein and simply write the name of the protein. Suppose I want to study hexokinase. Then type in hexokinase. Okay. And see the various forms opens up. The hexokinase of Drosophila, Ossophylla, everything opens up. Suppose I want to understand the this one. Okay. Or you can get to see from here also you can get to see the FASTA. Okay, the FASTA means the protein sequence. For everything you can have the FASTA. So simply click on FASTA, and once you click on FASTA, then the entire protein opens up. So if you click over everything, then you get the FASTA format. If you only want the amino acid, then you can simply copy it and copy it. Okay, so that's how you can get the protein structure
Okay, sir.
sequence. Okay.
Yes, sir. I
Okay.
got it.
Okay. Sure. Okay. Any other problems?
A question I have,
Yeah.
not regarding the topic, I understood what to do. Sir, will you provide the video? Because I think yesterday's
Yes,
video
yes.
is.
I'm going to give you yesterday's and today's class, both the
Okay,
videos.
sir. So, that will be easy for me.
Yeah,
Maybe
yeah,
I just.
actually, I couldn't actually I forgot the password actually for the
Okay.
email ID, so I couldn't load it yesterday. So I'm going to load it today. Okay, upload it
Okay,
today.
sir. Okay, sir. So then just the work will be just creating this type of a structure of a protein, right? So that will be a practice.
Yes,
Okay,
creating a structure of the protein and analyzing the structure.
okay, sir.
Okay.
So suppose I'm
Hmm.
analyzing this structure.
Yes.
So what I need to write, like in order to make a clear analysis on a report.
To make a clear analysis, what you can do is you can see the electrostatics first, the vacuum electrostatics. Then, when you are modeling a protein, try to do a literature survey to understand what are the most important residues that are there that are required for a particular protein's interactions. Okay, and suppose you find out that these two residues or this domain is actually very important for protein blocking and other part or binding to a other protein, or it is very important in the activity of the particular protein. So check that, remember or check that particular domain and try to analyze and find out where that particular domain is located. Is it located, is it showing a ligand docking site? Is there any crevice there? Because if there's a crevice, then obviously it's a part of a ligand binding site. Okay, and suppose in any case, suppose your particular domain is buried inside, then what? Then you know that it is not giving you a structural advantage, but it is modulating the structure of the protein from inside. Okay, also
Okay,
you
sir.
get to know, suppose there is a lot of you know that suppose you have a protein with which is highly electronegative. Now, obviously, if it's a protein with high electronegativity, then you know for sure, not for sure, but you know that that particular protein has something to do with DNA binding. Okay, that has something to do with DNA binding. So these are the kind of analysis that you can do. But obviously, it actually depends upon that which proteins are you selecting, which protein are you selecting. Because when you are selecting a protein, then depending upon its function, there are analysis.
Okay, sir. So basically, the main points or the common points will be the electronegative domains
Yes,
and
yes,
electropositive domains
yes.
and the ligand binding side and the
Right.
structure.
These are the common points. And to
Okay.
make a more detailed analysis, you have to actually look into the function of that particular protein. That in this particular protein is responsible for which function, and depending upon the function, you can relate to the structure. Because we know the SAR. What is SAR? SAR is structure activity relationship. So depending upon that, you can actually go back and then analyze the structure.
Okay, sir. Like why that protein is
Yes,
performing that function.
how can this structure help to attain this function?
Okay, got it.
Okay, okay. Any other queries?
No, sir. I need to perform once.
Yeah, yeah, sure, sure. Go ahead. Do one thing. I'm uploading the videos, so you perform this, and then if you have any problem, get back to me. Okay.
Okay, sir. Like if I have any problem, I will just drop the screenshots and the doubts.
Yeah, yeah, sure, 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 sure.
Okay, sir.
Okay. So by then,
Okay, sir. Thank you.
sure, sure. Thank you.